We discussed proteins, and now we are going to limit our discussion on a specific type of proteins, which are enzymes. The objectives of this video are to understand the function of enzymes, which is speeding up chemical reactions, and to understand how enzymes do this function of speeding up chemical reactions. So let's begin. Enzymes are globular proteins that act as catalysts. They have a globular shape, they are proteins, and their function is to catalyze reactions, which means they alter the rate of chemical reactions without eventually changing. So the enzyme leaves the chemical reaction having the same globular shape. And they can be reused rapidly and repeatedly, which makes them very useful and efficient. Once an enzyme finishes catalyzing the reaction in one place, it can directly go and do the same in another place. Let's take this chemical reaction, the hydrolysis of sucrose into fructose and glucose. Sucrose and water are what we call the reactants. They are what we have initially and what are going to participate in the chemical reaction. Fructose and glucose are what we get after the reaction is done. So these are the products. So we have reactants and products. For this chemical reaction to occur, some conditions need to be met. They can't just occur out of nowhere. The first one is that the sucrose molecule and the water molecule must be colliding or actually physically bumping into each other with a high enough energy in order to allow them to break bonds and make new bonds. The water is going to break a bond, the glycosidic bond, and the hydrogen is going to insert itself, and then OH is going to insert itself. We saw this in the hydrolysis reactions of disaccharides. So sucrose and water must be colliding with each other at a high enough energy. That is the first condition. The second condition is that the free energy of the products, which is how much energy they have in their bonds, must be less than the free energy of the substrates. And this is a concept in physics that can be hard to grasp, but simply put, this means that the reaction can occur spontaneously, or in other words, it does not need an energy input, like for example heat. Some reactions need to be given heat as a form of energy, or else they cannot occur, simply because the products have a much higher energy than the reactants. So you need an extra input of energy from somewhere. For this reaction to occur spontaneously without an input of energy, the products must be lower in energy than the reactants. And the third condition is that there is a minimum amount of energy that has to be present in the beginning for the reaction to start. So there is a bit of an energy barrier at the beginning, which is what we call the activation energy barrier. The activation energy barrier is the condition which the enzymes help in. So this is the graph showing the free energy as a function of time. This is the beginning of the reaction, we have the substrate, and this is the end of the reaction, we have the products. The substrate is higher in energy than the products are lower in energy, so this is one condition that is met. The substrates are colliding with each other, but there is an activation energy barrier. They need to have high enough energy in collision in order to cross this barrier and have the reaction start. See here, this first line, how high it is. There's a very high activation energy barrier. And this is what the substrates have to achieve in order for the reaction to happen on its own. And this is never going to happen significantly. This is going to be a very, very, very slow process because the substrates have a very high activation energy barrier. What the enzyme does is here we can see in the presence of the enzyme, it lowers the activation energy barrier. So now with the enzyme, the activation energy is much lower, almost half even, and the substrate can cross this energy barrier comfortably now and fast enough in order to get to the products for the reaction to take place. And this is the function of enzymes. They lower the activation energy barrier for the substrates for the reaction to take place. Again, this is the reactant, and this is its energy barrier when we don't have enzymes. Here is the energy barrier when we have enzymes. You can see the difference. The reaction on the left with no enzymes is going to be very, very, very slow and inefficient because there is a very high energy barrier, while the one on the right with the enzyme is going to be fast, it's going to be efficient and rapid because there is a very low activation energy barrier.